This session is going to be looking at the PPIE component of the EJS Act project, and the co-chairs of the PPIE working group for the project are Dr. Kevin McFarthing and Michèle Bartlett. And I think we'll start off firstly just by asking both to introduce themselves. Ladies first, Michèle.
All right, I'll go first.
Ladies first.
I'm originally a biochemist, which when I was diagnosed with Parkinson's was a very strong asset. I could understand a lot of the language that was being used to describe the science of Parkinson's. Since then, I'm on the research committee for Cure Parkinson's, on the editorial board of the Journal of Parkinson's Disease, and one of the authors of an annual pipeline review, clinical pipeline review, which you saw earlier in Harris's talk. I also curate what's called the Parkinson's Hope List, which is a database of all the projects that are going on in the research stage, discovery, preclinical, and the clinical phases of phase 1, 2, 3. And there's almost 400 different projects on that database. I occasionally get exposed to fifth-year medical students at Oxford University as an expert patient tutor. So they get experience of pushing, prodding, twisting, various bits of my arms and legs to understand what it's like to see a real person with Parkinson's. And I was diagnosed with Parkinson's at 4 p.m. on Monday, the 10th of December, 2012.
Thanks, Kevin. So I'll start the other way around. I was diagnosed in December 2017, so I'm coming up to my 8-year anniversary now. I'm a psychotherapist, so I'm very interested in what's going on in people's heads in a completely different way to the neurologists. I've been really interested in being involved in research ever since I was diagnosed, really. I was a participant of the exenatide PD3 phase 3 clinical trial. I'm in some long-term observational studies. I am, as well as being involved in the PPIE working group for EJS Act PD, I am a Cure Parkinson's research advocate, in which capacity I attend the International Clinical Trials Committee and also the International MAMS Consortium that Shireen was talking about. And I am a co-chair of a patient advocacy council for the critical path of Parkinson's.
Wonderful. Okay, thank you very much. The first question, before we open up to questions from the audience, we'll just go through a couple of warm-up questions for you. So how did you find out about EJS Act PD, the project itself, and what aspect of it did you find most interesting? What drew you in?
I actually can't remember, but I'm pretty certain it was through probably Cure Parkinson's or Parkinson's UK, because I'm part of their research support network. And I thought it sounded really interesting, partly because as a participant of a phase 3 clinical trial, I was interested in thinking about how we could make trials more participant friendly, how it would be possible to contribute to that. The thing I specifically remember, and I don't know how it's phrased, Georgia might remember, is there's something in the application that implies that they wanted people who were quite opinionated and not afraid to speak up. And I thought, yeah, that's me.
Bullshit.
Bullshit.
It was fairly simple. I got an email from Professor Camille Carroll asking me if I'd like to chair the group. So
why might someone else consider joining the working group as opposed to getting involved with the clinical trial itself?
I guess so my particular hobby horse is outcome measures or endpoints and how we measure whether something's effective or not. Because I don't think, you know, Harris, I think, said it earlier when it was, if you had $1 billion, what would be the thing you'd go for right now? And it would be, yes, can we have one measure that works, that shows that things work? Because, you know, all of these phase 3 trials carry so much hope, and then we can't show that things work because it's such a heterogeneous condition. It's so difficult. And so, yeah, my thing is outcome measures, and that's why I'm really pleased that we have patient-reported outcome measures in EJS Act PD, because I think it's, rather than going in once every 6 months and doing this, and all sorts of other things that aren't relevant to kind of our everyday life and what's meaningful to us, what's going to make a difference to how we can function on a day-to-day basis. For me, that's the measure of whether something works or not, and I think it's really important. So for me, being in the outcome measures working group felt really important, and I'm sure Kevin can. I think you have to find your niche really. What's the thing? If you wanted to join a working group, it's kind of what's the thing about research that's really important to you? What would you like to have an impact on? What would you like to influence?
I think it's the opportunity to contribute, to shift things even just a little bit, to make a difference to ultimately to people like us, this generation and future generations. So whether attending a meeting like this is helping to do that. The more people who attend meetings, the more people who are aware of potential for Parkinson's research, the better it is for all of us.
Wonderful. Okay, thank you. How was it actually chairing the working group? And knowing what you know now, was there anything you'd done differently if you were to start over? How was it, firstly, just chairing the working group?
It is very interesting because, as Michèle said, we deliberately asked for people who are prepared to voice an opinion. And so chairing the meetings was not quite like herding cats, but allowing different trails of discussion to freewheel it, if you like, whilst keeping an eye on the time, was quite a challenge. But fortunately, I had lots of experience in industry chairing meetings, which I think helped.
I'm thinking actually that I think I got off quite lightly because you chaired most of the meetings, and I would stand in for you sometimes when you weren't available. But I'm just thinking about the way the PPIE working group works together, and there are lots of strong views, but I think we all work in a very cooperative way. We all are aiming for the same thing, which is to, you know, initially when we all met up was just to get this initiative going. And it's been amazing to work on this project. It's been a privilege to be involved and to help shape the research, I think. And I feel like the whole PPIE working group, we do all have that common aim that we really want to make a difference. We want to shape Parkinson's research towards what's important to patients. We all want to find something that works, disease modifying or a cure. And so I think there's a very, and really also, I feel like we're really integrated into the whole of the consortium. And that is what I feel is really unusual about this. We've been involved since the inception of this project. We're completely woven through the fabric of the whole consortium, really. We're not a satellite, are we?
No.
There's no sense of us being there to rubber stamp something, you know. We've been able to really make a difference. I think we all feel that. So although, as you say, Kevin, sometimes you've got very disparate views on something, and some things generate a lot of heat, but it's important to have a wide range of views so that we can represent the Parkinson's community.
Yeah, as Michèle said, the word shape is important. We've been able to shape the big picture at the same time as scrutinizing the detail. So things like the patient information sheets will have been co-written and approved by the PPIE group. So there's lots of output from a clinical trial, not just the results. But almost every bit of it will have been through the patient and public involvement and engagement group.
And just coming back to the first part of the question, second part of the question, if you were to go back and start all over, is there anything you'd have done differently?
Ooh.
Just so everybody in the audience knows, that question is not on the script, so these guys are not prepared for it.
Maybe to have a bit more training at the start,
Training.
because the clinical trials industry is acronym heavy. So if you're experienced in it and you're knowledgeable about it, these acronyms are straightforward. But to somebody who's naive to that area, it can be very confusing and potentially dispiriting. So I think I would say more preparation up front.
I was thinking the same thing, actually, Kevin. I was thinking, I think I would have tried to prepare myself better and be more aware of what was involved, and prepare myself in terms of, you know, my knowledge base perhaps a bit more. But I feel like the project has really helped us. I mean, we've had forums, haven't we, on different topics. So the nuts and bolts of things like treatment selection and, you know, how the statistics work, how you work out how many participants you need for powering a trial and so on. So we, you know, all of these things that are quite scientific and technical, if for people that aren't coming, unlike Kevin, who comes from a scientific background, but who perhaps don't have an understanding of how all of those things work, we've had a lot of support around, but perhaps we were playing catch up at the start, I think. And I think that learning's gone into some of our international the international MAMS trials, thinking of sort of more upfront about how they can resource the PPIE working groups before they start.
Well, on the plus side, we have what our accountant colleagues would potentially call an intangible asset. That's a group of people who are now very experienced at going through the development of the clinical trial, which will be very beneficial for the future of this project.
Fantastic. I'm going to open up the floor now for questions. If anyone has any questions, please raise your hand. Rachel, up the back there.
This isn't quite exactly related to this that we've just had, but I think it was the first talk, and on the screen there were two organic molecules of cinnamon and coke cardopa, is it? And I'm just interested because if they run out of Cinemet, then they usually give me that. And I can't, if it's different organically, does it work the same? So if a trial, would you ask them to be on the same Cinemet, or does it not matter?
So if I get this right, Rachel, he's saying, is it someone is switched from Cinemet to Madapar, for example? Does it make a difference?
If Cinemet is like the stocks are low, and looking at the organic picture, they don't look the same. So how does that work on the trial?
The levodopa that you get in co-caroldopa or Cinemet is exactly the same organic molecule.
Same.
Yeah, so there might be some differences in what's called the excipients, the other ingredients in the tablet. But they should have gone through the testing to show, at least in the laboratory, an equivalence of dissolution rate and things like that.
But same, I think they work differently, even though they're supposed to be the same. It's the same as any like new drug in there. And then when they lost their patency, they seem to work differently.
Right.
Well, there's no doubt that generic versions of branded drugs can have different effects in different people. And often people, you know, get upset if they've been on Cinemet for years and suddenly the pharmacist gives them co-caroldopa and they say it doesn't work as well for me. And the opposite can occur. Someone that starts on co-caroldopa can get upset when suddenly they're given Cinemet. So it's just what people get used to over the years. But as Kevin said, the drug is the same, but how quickly it's released and what its peak may be in the bloodstream in the brain may differ, and so people experience different effects. But you know, that's not something we're taking into account in the trial. You know, everybody has their optimal levodopa or other drug treatments throughout the duration of the trial, and what we're looking for are things that change despite the symptomatic agents that people are given.
Any other questions?
Do you feel that we've got the right balance between the basic chemistry, the research into chemistry, and trials? It seems to me we could be very one-sided on the trials side, I have a feeling there's more to be gained working on the base chemistry.
I think looking from the data that I have in the hope list, there are many more projects in the research stage looking at discovery, and basic chemistry would be part of that. Because the pharmaceutical pipeline is often presented as a funnel with lots of options going in at the start, and then narrowing down to a small number of options. So there is a phenomenal amount of work being done on the basic biology of Parkinson's, trying to understand what goes wrong, and looking at potential ways to correct it. And that's something that obviously we should encourage. But I think the balance, if anything, might be moving away from the clinical side, given that we have, I think, 6 phase 3 disease-modifying studies underway. And 2 of those are coming from the AGS ACT-PD project, and with a 3rd one coming on within the next year.
Sorry, question here.
Hi, this question is sort of related to PPIE engagement, and more probably for the researchers in the group. So the UK population's got so many communities and ethnic groups. Have the researchers considered how to include these people in your trials? I know there is an MHRA, HRA consultation document on how we are more inclusive and diverse, but has this been considered by the researchers?
Yes, very much so. We want the trial to be as inclusive as possible. We want to have a representative cohort of participants, because often trial participants just tend to be very skewed towards a particular demographic. And we're very aware of some of the barriers to participation.
Yeah.
And in fact, there's the core PPIE working group that Kevin and I have been involved in, but there is a wider PPIE working group, or it's not actually, it's a wider group. What's it called? The PPIE network that we can draw on, who actually are drawn from a much more diverse type of range of people. And we also have the community advisory panel, who they're not all people with Parkinson's, but they all have long-term chronic degenerative, or care for somebody with long-term health conditions, so that they are giving us a much more diverse perspective on the decisions and some of the questions that we've had. And if we have a question that we feel the core group can't answer, then we put that out to the network or the community advisory panel. But we do also have a number of initiatives, and we've been working with our non-profit partners in thinking about how do we reach some of these communities that typically don't even know about research.
No.
And I think that's some of the problem. When you look at how many people with Parkinson's are involved in research, it's tiny. I think it's 1% of people with Parkinson's are involved in research. And that's not in clinical trials, that's involved in any way. And we want more people to be involved, because it's only by getting people to sign up to do the work, you know, either by participating or helping researchers to think about the questions, you know, and to design the research in a way that works better for people with Parkinson's. That's the only way that we can hopefully get this thing to go faster. But it needs to be a broad demographic of people involved, very much so. That's part of the reason we have so many sites all over the UK, as Georgia showed you on the map.
Yeah, that's interesting, and thank you for that. But how do you move forward if your patient information sheet, your consent form, your poster advertising your trial is all in English, and if English isn't your first language? I know the MHRA wants the documents in English. Now, I don't know how you do it in different languages. It's something that can be discussed, but I think it's quite important that it's considered.
We have considered that.
But does
Well, if-
MHR accept AI documents? I don't think so. They just accept English documents.
So there's, there's a major priority to make sure that we're being inclusive in this trial, and that extends to age and biological sex and geography and socioeconomic status. So we're taking the first part of the postcode for every participant, so we can look, because the first part of your postcode maps to your wealth. Different streets around the country are very representative of socioeconomic status. And of course, ethnicity, we're collecting data about ethnicity, and we know what the breakdown of ethnicities across the UK and which sites are likely to have access to people from different backgrounds. One of our limitations is that our primary outcome is the MDS UPDRS, and it's been validated in a certain number of languages, and so it's been tested that the questions capture in that language what we're trying to capture. It's tempting to use AI to translate to whatever languages, but it's got to be formally tested that translation captures what is actually hoped to be captured, and that hasn't been done in as many languages as there are people living with Parkinson's in the UK. So we can use the tools that we've got, and that does capture a lot of languages that are spoken in the UK. We've got some funding to make sure that we not only have targets, but if we're under recruiting from a certain demographic in central London or in Luton or Bradford, or if there are sites where there ought to be major recruitment within a certain demographic, we will make initiatives. We'll have champions that people will talk about their participation in the trial, and spread that message to communities that they come from, in order to make it more realistic, that people say, okay, this isn't just for young white middle-class men, this is for people of all backgrounds in the UK, and there are examples where people have had a positive experience and therefore perhaps I want to get involved as well. So we are really trying to prioritise this, and we recognise how important this is.
Any last questions? Please.
I really don't know a lot about this, so sorry if it's a silly question, but with the worldwide consortium, obviously there's lots of benefits to having lots of different people from all over the world be part of it. Are there any restrictions? Because maybe not for the UK, but are we holding anyone back, or is anyone else holding us back with what's available with all the regulators?
That's a really interesting question.
Sorry.
No.
I don't want to look at the negative of it, but I'm just wondering, it's great to all be together, but are there also things that could be keeping us back or keeping other people back?
So around the world, we've got projects like GP2, which are looking at underrepresented populations. They're going in and they're setting up networks, and they're just looking at collecting blood samples and looking at the genetics of Parkinson's disease. And while the genetics data is very interesting, the networking is really, really important. It's bringing people to research who are from these populations that have not typically been studied before. And that could be an avenue for future resources to go to clinical trials in those communities. At the moment, it's probably a resource issue more than a legal, or although maybe other people have alternative ideas on that.
I was just going to add, I think the most important thing that all of these studies can do is to make sure that their consent forms are aligned, so that you're getting as broad a consent as you possibly can, which then means that even if the physical data can't move, at least you've got permission to be able to dive into those data sets and answer critical questions. Because I think the thing that is so powerful about this, and going back to Shireen's presentation, we've got EJS at PD, that's 1,600 people in the UK. Shireen, how many people are will be in the master trial? Do we know yet? Harris, how many are going to be in Sleipnir and Hydra? Hydra in particular?
To start with
So that's 90.
40 times 320, but you know we will be adding things as we go.
Yeah.
Raise 200 times 4.
Yeah.
So 800.
So you can see if we've all collected the same data and the same data points in the same way and got the same broad consent and also are collecting the same biosamples, suddenly how powerful that is going to be, because we're really going to have a very large, very detailed set of data, and we'll really be able to understand the subsets of this disease, and with that, who is responding and why to what types of treatments, which is perhaps a bit simplistic.
Great question, though.
Very good question.
So if there are no other questions, I'm going to draw the session to a close. And I'd like to thank, firstly, Kevin and Michèle for taking part today.